And there's my recording. All right, you guys ready? Okay. All right, start my stopwatch, and here we go. Good afternoon. Our names are Dan, Josh, and Noah, and we're going to be going over Salmonella bacteriophages, specifically bacteriophage therapy. In recent years, a consistent problem with modern medicine is the repeated misuse and overuse of antibiotics in the treatment of disease. This trend has led to a rise in something called superbugs, in which bacteria are resistant to the antibiotic drugs we normally use to combat this disease. These superbugs can cause widespread illness, with examples such as MRSA and others that have been resistant to the antibacterials that we have used to try to combat them, and they've spread to numerous people throughout the hospitals where they have been contaminated. Bacterial resistance to to antibiotics emerged around the time when we started using antibiotics as a main method for combating disease, which occurred in 1928 with the discovery and implementation of penicillin. Penicillin rapidly became one of the most widely used antibiotics in the world, even to this day. However, because it has been used so frequently in numerous people, it has become an issue because it selects for antibiotic resistance. By killing off all the bacteria that are resistant, that are not resistant to the drug, but letting those that are survive, it selects very heavily for antibiotic resistance. Now, even with our wide array of antibacterials in use in modern medicine, there's even stronger selection for antibiotic resistance. So a potential solution is phage therapy, in which bacteriophages are used to combat these superbugs. This has become an increasingly promising method to combat antibiotic resistance because they are host specific, and these bacteria can be used to combat the exact form of the bacteria that is infecting a person without causing illness or danger to the person. With the use of host surface cell receptors to pick and choose exactly which bacteria to target, lytic bacteriophages could potentially replace modern antibiotics once the treatment has been refined a little bit more. And in reality, phage therapy is not even a brand new topic in modern medicine. Since they were discovered in 1915, there have been numerous efforts throughout history to combat bacteria-related diseases using bacteriophages. In modern Eastern cultures, such as Georgia and Poland, phage therapies are common treatments for bacterial infections. One example of this is the Phage Therapy Center in Elieva at the Elevia Institute in Georgia. Phage therapies are effective in treatment of antibiotic resistance diseases. However, they must be refined a little bit more in the future in order to get rid of any unwanted side effects, as they are prone to develop right now in current trials.
So bacteriophages are literally bacteria eater. That's derived from the name. And they were discovered in 1917 and 1919 by Twart and Durrell. And immediately upon this discovery, Durrell tried to use bacteriophages to treat dysentery. So that's like the first example of phage therapy. And they are highly diverse in both physical and genomic sizes, and um, and in conformation too. So if you look at this figure, the about 90% of the known bacteriophages fall into these three groups with Myoviridae, which are bacteriophages with long tail and contracting tail. Um, Cycloviridae are bacteriophages with long, non-contracting, and then Potiviridae are bacteriophages with short tail. And all of the phages discussed later in this presentation will be in these three. Bacteriophages are very abundant in nature. They are found anywhere there's bacteria. And so therefore, they are highly evolved and very host specific, and even in some cases, strain specific. So the bacteriophage life cycle starts with attachment and DNA injection. After that, the bacteriophage can choose between two life cycles, lytic or lysogenic. Life cycle is just the bacteriophage will replicate itself inside a bacteria and then lyse the bacteria to get itself out and infect other bacteria. The lysogenic cycle is a little different. The bacteriophage will integrate itself into the bacteria either as a separate plasmid or into the bacterial genome, and it'll get replicated every time the cell divide. So scientists classify bacteriophages into two classes: temperate bacteriophage that can do both, and the virulent bacteriophage can only do lytic cycle. So the life cycle starts with attachment. Bacteriophages usually have a very positively charged tail that it can attach itself to the bacterial membrane. And these are some of the typical membrane elements that it can bind to, like glycolipids or membrane proteins or even monoproteins like the flagella. And the molecular mechanism for DNA injection depends on the type of bacteriophage. Shown here are the mechanism for virtually all of the types of bacteriophage, and you can see that it's a little more complicated, and a lot of it is unknown, as seen in like the question marks right here. But all of them have in common is that there there has to be some host factors that assist with the process, and it looks like there is some digestion of the bacterial membrane involved. So with that, bacteria, with any arms race, bacteria have evolved to be phage resistant. And here are the 8 ways in which bacteria can become resistant to phages. I'm only going to talk about 2. One is inhibition of phage absorption. That's basically mutating the cell surface proteins so that the phage cannot bind. Or it can be just evolving a different pathway to coat the proteins in sugar so that the phage cannot bind. And also the cleavage of injected phage DNA. This is important, especially in molecular biology, because this gave us CRISPR-Cas9 and restriction enzyme. Restriction enzymes are like the primitive form of phage resistance because it just recognizes palindromic sites in the phage genome and cut it up. And Cas9 CRISPR system is just more targeted in that there's a template of past phage infections that the bacteria can make RNA out of and use that RNA to guide the Cas system to cut precisely depending on the sequence.
So now I'll be talking about a specific paper on Salmonella bacteriophages just to exemplify the activity of bacteriophages. This paper was done in 2017 by Jung et al., where he and his team attempted to combat antibiotic resistance in Salmonella bacteria using bacteriophages. He analyzed 6 different bacteriophages and used those bacteriophages to infect 4 different Salmonella bacterial strains, including an antibiotic resistant strain of one of the Salmonella bacterias. So we'll just be going over a few of the experiments in order to highlight the ability of these bacteriophages. So in one of his experiments, he wanted to exemplify the problem of antibiotic resistance. Looking at, each column represents one of the 4 Salmonella bacteria, and focusing on the 2 middle columns, you can see that one of them is Salmonella ATCC 19585, and the other one is ATCC 19585 ciprofloxacin induced. This is just the antibiotic resistant strain of the ATCC bacteria. So when comparing the 2 columns, you can see that the non-antibiotic resistant strain was susceptible to almost all of the antibiotics that the study looked over. But comparing it to its antibiotic resistant form, many of the antibiotics that it was once resistant to or once susceptible to, it became resistant to those, as indicated by the blue rectangles. At another experiment, the study infected one of the Salmonella bacteria called KCCM 40253 with all of the bacteriophages at increasing multiplicities of infection. Firstly, you can see that the bacteriophage PT2B1 had the lowest mortality rates against the bacteria at all multiplicities of infection, but the bacteriophage PT2 had the highest mortality rates at all multiplicities of infection. This could show that PT2 could be a good candidate in dealing with the Salmonella bacteria infections, and also shows that the efficacy of lytic activity correlates between the ratios of bacteriophages and hosts, meaning that the more bacteriophages that are present, the more likely the bacterial hosts are to be lysed. In this experiment, the researchers infected all 4 strains of Salmonella with all 6 bacteriophages. Firstly, you can see that the Salmonella bacteria CCARM was not able to be lysed by all of the bacteriophages. One conclusion that the paper went over was that the CCARM strain had altered receptors on its bacterial surface. So meaning that the bacteriophages may not have been able to recognize the receptors and thus were not able to engage in its life cycle and lyse the CCARM strain. Going over the same experiment, we can see that PT2 is able to lyse the other 3 bacterial strains, and that when comparing ATCC to its antibiotic resistant form, it's able to lyse both strains at relatively equal proportions, meaning that PT2 may be a viable candidate for killing the antibiotic resistant strains of Salmonella, and that antibiotic resistance does not affect the phage's ability to lyse that bacterial host. So the overall take-home message is that bacteriophages are possibly the solution to dealing with antibiotic-resistant bacteria. This is exemplified in not just Jung et al. Study, but in several studies concerning phage therapy. But definitely more research needs to be done before bacteriophages can be fully accepted in the realm of medicine. Some questions that were left unanswered were, as antibiotic resistance of bacteria is becoming a problem in defending against illness, will phage therapy be the next step in medicine? Again, more research just needs to be done before we can start using this in modern medicine. Another question is that, can new variants be produced to lyse CCARM Salmonella strain? In the previous experiment, CCARM was not able to be lysed by any of the 6 bacteriophages, but possibly an undiscovered variant or a variant that was not used could be used to lyse that bacterial strain. And another question is, will PT2 affect the good bacteria within the human body? Just based on the paper, it's unknown if PT2 will affect the human microbiota, so definitely more research needs to be done. And that is the end of our presentation. Thank you so much for listening.